Okay, folks, we'll go ahead and get started again. I'm Simon Swanberg. I co-organized this seminar series with Bilal and Garrett. So just a quick announcement before we get started. We have several more of these before the end of the semester, but Bilal and I are already starting to think about who we might invite in the next year and in the coming years, and we're sort of keep a running list of things. So one of the things we've been wanting to do is reach out to people to get more suggestions for people to participate in this seminar series. So if you have recommendations, I would just encourage, we can even do potentially international people now through these virtual ones. So we're discussing how to go forward with this in the future. But if you have any speakers that you would really like to see come to speak at Georgia Tech, either virtually or in person, because we will be picking up in person, hopefully by the fall, please send us an email and let us know. We'll send out a more formal survey in a little while like we did last year, but I'd like to just have an open invitation for people to contact us. Also, I'll just remind everybody that at the end of this seminar series, the end of today's talk by Hannah, we'll be having questions, and I would like the first question to come from a student or a postdoc. So we'll give a little bit of a pause at the end. So if you're a student or a postdoc and you have a question near the end of the talk, please let me know. You can write it in the chat and just indicate that you're a student or a postdoc, or you can just raise your hand and I'll call on you. Okay, so we'll go ahead and get started. It's my pleasure to introduce Hannah Choi. Hannah just joined us from the University of Washington. She's a new assistant professor in the School of Mathematics, but is also really involved in computational neuroscience and the neuro community. So I want to just take this moment to formally, I guess, welcome Hannah again to Georgia Tech and to our community. It's really nice to have you here. I'm really excited to see all the cool things you and your collaborators can do here. So Hannah comes from an applied math background. She did her PhD in applied mathematics at Northwestern. She then did her postdoc at the University of Washington, where she was a Washington Research Foundation Innovation Postdoctoral Fellow in Neuroengineering, which is quite a mouthful, but it's a very cool program for neuro postdocs at the University of Washington. For any of our graduating PhDs, you may want to look at that program. She was also a Simons Berkeley Research Fellow and a McGovern Research Fellow at the Simons Institute for Theoretical Modeling for the Theory of Computing. And that was at UC Berkeley, where she was a visitor for a semester in 2018. So she now joins us. She already has been awarded an NIH Pathway to Independence Award at K99. So she's off to a great start, and her lab is just getting started. So welcome, Hannah. As you guys know, we use this sort of seminar series as a joint thing to bring in exceptional external speakers to talk to our community and to highlight our young and continuing faculty members in the new research that they're doing. So Hannah, go ahead and take it away.
Thank you so much, Simon, for the very kind welcome. Yeah, so as Simon mentioned, I just started at Georgia Tech 3 months ago, and so I'm really new here, and I'm really excited to meet the neuroscience community. So I'm really happy to have this opportunity to meet everyone. So my research focuses on linking computational functionality and connectivity structure of the neural networks at multiple scales. And I look into both biological neural network and neural networks. And today I'll specifically focus on the hierarchical organization of the brain network and its computational implications. Right. All right, so I am being a little ambitious. I like to talk about three different things in this talk. So first, I'll start by constructing a structural hierarchy of mouse cortical inflamming network based on anatomical connectivity data, which was just recently released by the Allen Institute for Brain Science. Then I'll move on to this computational implication of the hierarchy structure of the cortical network. And specifically, I'll discuss the model of hierarchical predictive coding. Finally, if time permits, I'll introduce an ongoing effort on testing predictive coding theory in mouse visual. All right, so so I'll first tackle this first question: is the mouse cortical network organized enough? So in order to do that, we developed a data-driven, unsupervised method to uncover the hierarchical. in many previous studies, the brain network has been represented as just a simple graph. And for example, the Allen Mouse Brain Connectivity Atlas here is a good example of representing a brain network as a simple. So in this data, so this data provides a high quality and open source anatomical data for detailed, weighted, and directed network of the mouse brain regions. So in this data set, to construct a mesoscopic brain connectome, viral injections were, use my cursor here. Yeah. So the brain, so the viral injections were made at multiple locations in the brain. And the fluorescent tracer expression provides us this information which region is connected. So, so based on that, based on the fluorescent tracer expression, we can actually build this simple representation of brain network. Brain network based on this box of based on the fluorescent voxels and represent the mouse whole brain as a directed and weighted network of brain regions. So so this simple representation of the brain network already reveals some features of the brain connectivity, such as its modularity, for example, or but we can only get a partial understanding of the brain network from this single network of the mouse brain connectome. Of course, this is too simplified. It doesn't really represent like in the brain network. Even though it's pretty anatomically precise in all right, so in fact, when you look at this pair of brain regions, for example, this pair of brain regions within the network, and the connections between them, you shouldn't actually just simplify this as a single directed and weighted connection between these two homogeneous nodes, because this is not exactly what's going on in the brain network. Instead, what really happens in the brain network is that each cortical region has this laminar structure, often composed of 6 layers, with different types of cell populations in different layers in its cartoon. And among these layers and different cell populations, there can be multiple types of connectivity patterns. So for example, let's say we are looking into a connection from region A to region B, then it can have a connectivity that looks like this or that. And if you look into a connection from region B to region A, or so, it can depend on, so the connection should depend on the layers and cell types. So there can be actually a diverse connectivity patterns between two regions in the brain network. So the actual brain network should look more like this, featuring multiple types of connections between a pair of regions. And these connection patterns should be differentiated. So like, for example, this gray connection patterns compared to this yellow connection patterns, because they are different. And so such differentiation of the connection type is needed in order to get more complete understanding of the brain network. And luckily, the Allen Mouse Brain Connectivity Atlas was recently expanded to include structural connectivity data from pre-transgenic mice with cre-dependent viral tracing experiments. So what does that mean? Because different cre-dependent tracers can selectively label different cell types located in different layers within the cortical column, as shown in this beautiful picture of layer selective labeling of pre-expression. We can construct a cell type and layer-specific connectivity brain. So with this more complete picture of the mouse brain network, what can we learn about the network? So what we are doing here is to leverage this layer-specific connectome and to reveal another important structural feature of mouse brain network, namely its hierarchical organization. So the hierarchical organization is indeed extensively studied and is relatively well defined in the primate cortex, as shown in this illustration adapted from the seminal paper by Thalman and Darasin. And furthermore, the laminar connectivity patterns that define the directionality of connections primate cortex is well established. For example, in primate cortex, we know that the feed-forward signals tend to originate from layer 2-3, the shallow layer, and tend to target the granular layer 4 in the middle. And feedback signals tend to originate from the infragranular layer, so the deep layer 6, and tend to target layer 2-3 in layer 5-6, while avoiding layer 4, the granular layer in the middle. However, we do not actually know whether such rules exist in the mouse cortex, and we don't even actually know whether the mouse cortical network is hierarchical at all, as in the primate cortex. So here we ask these two questions. First, about the mouse cortical network. First of all, are cortical areas in the mouse are so organized into. And second, can the anatomical patterns of connections define the order? So based on the Cree-dependent tracer data from the Allen Institute, the team at the Allen Institute found various termination patterns in the target cortical layers in both cortical and thalamocortical. And using unsupervised clustering algorithm, these patterns, so the target layer patterns can be clustered together, yielding these 9 different clusters of different projection lamination patterns for both cortical and thalamocortical connections. So for example, cluster 9 here includes the connections that target, as you see, deep layers, layer 5 and 6 mostly. And for example, if you look at this cluster 6, this cluster includes projections that terminate in both superficial layer, so layer mostly, and in the deep layer 6, and so on. Okay. So here is a schematic of this classified different connection types depending on the cortical layer termination pattern. So here is this nine cortical layer termination patterns that we clustered, and we then investigated whether these patterns can define the directionality of the connection, namely whether it's feed forward or feedback, as in the primate cortex. All right, but the problem is that we don't really know whether the same anatomical rules from the primate cortex defining each lamination pattern as either feed forward or feedback is relevant here. And we don't even know whether such rules exist in the mouse cortical network. So we implemented an unsupervised method to find the mapping between the layer termination patterns, so there's 9 different patterns, and the direction, namely feed forward or feedback in the hierarchy. So what we are doing here is to map this connection pattern, so 9 different connection pattern, to either feed forward or feedback. So this is to find so if you find that mapping as M, that's to find the mapping from this 9 different patterns, 1 to 9, well I said C here, to either plus or minus 1, feed forward or feedback. And to find the optimal mapping, we search through all maps between the layer termination patterns, and that's 9 different patterns, and the direction, plus or minus 1, to find the most self-consistent hierarchy. So what do I mean by most self-consistent hierarchy? First of all, we define the hierarchical level of each brain region as the average direction of connections to the area minus average direction of connections from the area. So let's say we are looking into brain region I, and then its hierarchical level is defined as H sub I, and and that can be defined as the average between the hierarchy of region I as target, which is the first term, and the hierarchy of region I as source, which is the second term here. All right, so let's look into this a little more closely here. So here, C sub Tij, so T, we don't really need to worry about T here. So C sub Ij defines the connection patterns from region J to region I. Okay, so that's why I'm saying that this is hierarchy of region I as a target. So the C sub Ij can be one of these 9 different cortical layers. And this mapping M of C sub ij maps this 9 different patterns into either plus or minus 1, depending on whether that pattern corresponds to feed forward or feedback connection patterns. Okay. All right, and yeah, so here the first part basically, and then that mapping is averaged over all different J's, where the connection, which actually send the connections to region region I, and similarly here, the second term defines the hierarchy of region I as source, because we are mapping which is this M. We are mapping this connection pattern C sub ji. So this is a connection pattern from I to j, which is one of these 9 numbers, 9 different cluster patterns. And with this mapping M, this number is either plus or minus. So if the region I gets a lot of feed-forward connections and receive a lot of feed-forward connections and send a lot of feedback connections, then that means that actually increases the hierarchy level of region I. And if the region I receives a lot of feedback connections while sending a lot of feed-forward connections, then this value h sub I will be low. And put the region in like somewhere lower in the hierarchy. Okay. All right. So and then based on this, we can define this global hierarchy score, which is a measure of self-consistency of the obtained hierarchy with the directions of individual connections. So we define that as H. And which is basically M of C sub ij times H sub I minus H sub j, averaged over all regions I and j. So what does that mean? So let's say in the obtained hierarchy, region I is higher in the hierarchy compared to region j. Then H sub I minus H sub j should be some positive number, right? And if that's consistent with the mapping, then that means then this value, so this value basically defines the directionality of the connection from J to I. That should be feed forward, and that therefore this should be plus 1, right? So plus 1 times some positive number will overall increase this value, the global hierarchy score. On the other hand, let's say they are inconsistent. So let's say in the defined hierarchy, region I is higher compared to region J, and therefore H sub I minus H sub j is some positive number. But if this is inconsistent with the mapping, then this value, so for the connection from J to I will be defined as feedback, and therefore this will be minus 1. So minus 1 times some positive number will be some negative number, and therefore overall that inconsistency will decrease the global hierarchy score. So that's how we define the self-consistency of the obtained hierarchy, and we basically search through all maps between layer termination patterns in the direction to find the mapping that maximizes this global hierarchy score. And by doing so, our unsupervised method, which simply maximizes this global hierarchy score and maximizes the self-consistency, group these 9 different patterns into feed forward and feedback. So according to our algorithm, for the cortical mapping, our method grouped layer 2, 6, and 9 as feedback patterns, and the rest as feed forward patterns. And for thalamocortical connections, our method grouped connection type 2 and 6 as feedback, and the rest as feed forward. And what you see here is that it's not exactly the same as what we see from the primary cortical network, but there is some sort of similarity. As you can see here, for the feedback connections, the projection tend to target either the shallow or the deeper layer while avoiding the middle layers. And for the feed-forward connections, a lot of connection patterns include the projections that target somewhere in the middle. So it seems like there is some consistency with the primary cortical network. All right, so by maximizing the self-consistency this way, and we can concurrently find the mapping rule and the hierarchy of the brain regions. But and then in this way you can find the initial hierarchy of the brain region. However, this initial hierarchy simply counts the number of feedback and feed-forward connections each area makes, and therefore it does not take account of the current hierarchy scores of its target and source regions. So to illustrate this point, if there are 3 professors reporting to a department chair, and if there are 3 undergrads reporting to a grad students, our method here will place them at the same level in the hierarchy. But that doesn't give us the full picture of the hierarchy. So we therefore wanted to adjust the hierarchy scores of the brain regions based on the hierarchy levels of its targets and sources. So if you're receiving feed-forward connections from a lot of higher areas, then you're gonna get moved even up. You're gonna be bumped up even higher in the hierarchy, and compared to the regions that receive a lot of feed-forward inputs, but from from the regions that are lower in the hierarchy. So that's how we iterated the hierarchy scores. So hierarchy scores are iterated until they reach fixed points, and that gave us the final hierarchy of all the brain regions. All right, but there is this fundamental question: are the mouse brain areas organized into a hierarchy at all? Maybe mouse brain network is not hierarchical after all. Maybe it's just some flat network. So to test this, we've compared the global hierarchy scores of the mouse brain data to those of an artificially generated, perfectly hierarchical network, as well as those of the shuffle network. So this actually shows the global hierarchy scores of the shuffle network here compared to the global hierarchy scores of the mouse brain network. And and here the green color denotes the global hierarchy score generated by including just the cortical connection data. Magenta is when we included thalamocortical connection data in addition to the cortical connection data. And blue here includes basically all the available data, so cortical, thalamocortical, and cortical thalamic connection data. Okay, and what we see here is that.
Can I ask a question?
Yes.
Why is the random or the shuffled data in pink, which has, in between green and blue effectively, why does it suddenly have this sort of shuffled large amount of hierarchy detected compared to blue and green?
So answer is I actually do not know exactly. In this case, what really matters is the difference between the shuffled and the non-shuffled, the data-driven connected data. So if you just look at that, that's not really different between green and magenta. But I guess for blue, it actually increases a lot.
I see. So like when you add in the TC connections, then it just shifts up, and then when you add in the connections, it shifts broader.
Yeah.
Thank you.
No problem. Thanks. That's a really good question. But what this shows in summary is that compared to the shuffle data, you see that the mouse brain regions have like significantly higher global hierarchy score, meaning that mouse brain network is significantly more hierarchical, because its hierarchy is very self-consistent. And also, as we see here, magenta and blue have higher global hierarchy score compared to green, meaning that the additional data on thalamic connections increases global hierarchy score, making the mouse cortical thalamic network more hierarchical. Okay. All right, so now we know that mouse brain network is And based on this unsupervised mapping that optimizes the self-consistency followed by the iteration scheme, we can now construct the hierarchy of cortical and thalamic regions, as we see here. So here we see the hierarchy of the entire mouse cortical and thalamic regions. And here, the different color denotes different cortical modules, and the gray denotes thalamic regions. And what we see here is that we see some thalamic regions at the bottom of the hierarchy, and some thalamic regions in the top of the hierarchy, and some interspersed in between. And when we look into the cortical regions, we see that on the bottom, there are a lot of the primary sensory cortical regions at the bottom of the hierarchy, and a lot of association areas at the top of the hierarchy. And then we can use exactly the same method to find the hierarchy between different cortical modules. And this is what we found. So for intermodule hierarchy, our method, our unsupervised method, which simply maximizes the self-consistency of the hierarchy, places auditory, visual, and somatomotor modules at the bottom of the hierarchy, while putting the prefrontal module at the top of the hierarchy. And when we look into intramodule hierarchy, so the hierarchy within each cortical module. So for example, when we look into the visual module, we see that our method places the primary visual area at the bottom of the hierarchy, while placing other like higher visual areas at the top of the hierarchy. Okay. So it seems like our unsupervised method, which depends only on the connection, like the layer-specific connection pattern, actually can uncover the hierarchical structure of the mouse cortical and thalamic network, which actually agree with our hypothesis. And recently, we compared this anatomical hierarchy to functional hierarchy obtained from neuropixel data from visual cortical and thalamic area. So a functional hierarchy could be obtained using various methods when you look into the electrophysiology data from neuropixels. For example, by looking into delays of the peaks of cross-correlation between different regions, or you can look into the time to first spikes, where you can look into response decay timescales. So these are all so on the y-axis, these are all some sort of measure of functional hierarchy obtained from the electrophysiology. And on the x-axis, we are comparing this functional hierarchy on the y-axis to anatomical hierarchy scores obtained from our unsupervised method. And we see that the correlation is really good between the functional hierarchy and this anatomical hierarchy, indicating that this uncovered anatomical hierarchy agrees very well with the functional hierarchy obtained from electrophysiology. All right, so we now know that the cortical network has a hierarchical structure even in the mouse. But what does this hierarchical network imply in terms of computation? So it actually implies a lot of different things, but I will discuss the theory of hierarchical neurocoding in this case, and therefore bridge the hierarchical architecture of the brain network and its computational implications next part. All right, so specifically, we'll focus on the hierarchical computation in the visual cortex. So in the primate brain especially, there have been a lot of studies on how the visual information is processed hierarchically, at least in the feed-forward way. So object recognition, for example, is processed in the ventral pathway, which is organized in a hierarchical fashion, starting from the primary visual cortex, primary visual cortical region V1, that where the neurons are known to encode very simple visual features such as orientation bars. Then it goes to V2, where the neurons encode slightly more complex visual features such as textures. Then there comes the intermediate visual area V4, where the neurons are for different curvature and angle. So they're kind of shape tuned. They can decode different shapes. And finally, there comes IT cortex, inferior temporal cortex, which is this higher visual cortical regions where neurons actually encode specific objects such as like face or inanimate face or cars. And prefrontal cortex, or PFC, is known to be involved in decision making and executive control behavior. So along the hierarchy of the visual cortex, yes. So it seems like we actually know a lot about the hierarchy of computation along the feed-forward direction. And in fact, many convolutional neural networks in computer vision, such as VGG, AlexNet, ResNet, and so on, are directly motivated by the feed-forward hierarchical structure of the primary visual cortex. And these artificial hierarchical neural nets have been very successful in object categorization, and some of them even matching or exceeding our visual system. However, to date, they are still not as robust as our visual system, especially when it comes to more complex visual stimuli or more noisy visual stimuli as we encounter in natural scenes. So for example, like a good example showing this feature is this like adversarial images. So this is from GoogleNet, another feed-forward hierarchical neural net for object categorization, where it actually categorized this picture very accurately as panda without any noise. But if you add some like really small noise, which is not like which to a human visual system doesn't really introduce as you can see here, then this GoogleNet gets totally confused, and now it actually fails to recognize what it is. Okay. So this actually indicates that there must be some additional complexity in the network structure unique to the brain, make it incredibly robust and efficient visual processor choice. And one of the obvious missing pieces in the convolutional neural nets is feedback connections. In many artificial hierarchical models are strictly feed-forward, while in the visual system there are a lot of. All right. So what is the computational role of the feedback signals in the? And one possible role of feedback has been suggested in hierarchical predictive coding theory, which proposes that these feedback signals carry prediction or some sort of internal expectation of the sensory stimuli. So in your brain, it has some sort of interrepresentation of the world, and it gets actively compared and updated. And in the hierarchy, in the cortical hierarchy, feedback signals tend to carry predictions about the lower area activities. And feed-forward signals are hypothesized to represent the residual errors between the feedback predictions and the encoding expectation. And in our case, we hypothesize that the predictive coding explains predictive coding explains the feedback signals in the visual pathway, and such feedback prediction becomes especially relevant when we have to process very challenging visual inputs, such as noisy visual inputs. So, for example, if the visual task is really easy, as if you are discriminating between these two isolated shapes, then maybe everything can be computed in the feed-forward way. But if it becomes a little more trickier, so if you have to discriminate between these two shapes under some occlusion, then maybe the feedback signals becomes very relevant. And then these feedback signals carry the prediction coming from the higher cortical area. And in particular, here we're going to focus on the feedback prediction coming from prefrontal cortex to V4 to explain the robust encoding of partially occluded shapes in area V4. So indeed, there is an experimental evidence for feedback signals coming from prefrontal cortex in V4 collected in Anita Pasupati's lab at the University of Washington. So in this experiment, the monkeys are trained to discriminate a pair of shapes on screen, so shape A and shape B, under varying degrees of occlusion, as you see here. And this is a delayed matching task. So the animal is first presented with an unoccluded reference, reference shape, and then after a little delay, the animal is presented with either matching shape or non-matching shape under varying degrees. And the animal has to decide whether the test shape is the same as the reference shape or not. And while the monkey was doing this test, recordings were made from both V4 and prefrontal cortex. And here is a single cell recording from an example V4 neuron to its preferred and non-preferred shape. So you see that V4 neurons are clearly shape selective as they respond strongly to the preferred shape, but not very much to this non-preferred shape, as you can see here. And let's look at the response dynamics to the preferred shape a little more. And here, different color traces represent different levels of occlusion. So as it goes from black to red, the level of occlusion increases, as you see here. And what we notice here is that there are actually two peaks in the responses. And during the initial peak, you see that the response decreases as the occlusion level increases. So it responds strongest to an occluded shape, and as it gets more and more occluded, the response goes down. And what's notable here is that there is the second peak of responses about 200 milliseconds after the stimulus onset. But if you look at this responses during the second peak of responses, you see that there are not that much difference between the black trace and the color traces, meaning that the response to an occluded shape is not very much different to the responses to partially occluded shapes. This is more clear if you look at the average firing rate during the first peak and the second peak. So during the first peak, the response decreases rapidly as the level of occlusion increases. However, during the second peak of responses, the decrease is a lot more gradual. So this indicates that the V4 responses are less sensitive to the occlusion during the second peak. So why do we see this delayed peak of responses in V4? It actually may be a result of the feedback inputs coming from higher areas like prefrontal cortex, PFC. All right, so here is a single cell recording from a representative PFC neuron. And what's really notable here is, first of all, this is single peak. And unlike in V4, PFC neurons respond strongly to partially occluded shapes, but only weakly to an occluded shape. So the response pattern is reversed in PFC. So if you look at the average firing rate as a function of level of occlusion, as you see here, as the occlusion level increases, the response increases, which is the opposite of V4. And furthermore, the peak of the PFC responses straddles in between the first and the second peak of V4 neuronal responses, indicating that the second peak in V4 responses may be actually induced by the feedback coming from PFC. So to understand the computational role of the feedback signals in V4 coming from PFC, we implemented the hierarchical predictive coding model of partially occluded shape discrimination composed of this simple two-layer. So the first layer is V4, and then the second layer is PFC. Okay. And in V4 layer, we included three different types of neural. So first one is shape A selective population, which responds strongly to shape A. And there is another class of neurons which respond strongly to some other shape B, but only weakly to shape A. And we have this third class of neurons which respond strongly to occluders. Okay? So it may be responding strongly to the color red of the occluders. These are connected with some connection weight matrix U to these two different neural units in PFC. And our hypothesis is that at V4, it combines the sensory inputs coming from the lower area, which have the information about the shape identity and the level of occlusion, and the feedback predictions made by PFC, which is represented as the weighted sum of the PFC responses, so weighted by this connection weights U with some nonlinearity F. At a V4, it integrates this top-down signals coming from prefrontal cortex and the bottom of observation of the sensory input and makes some hierarchical Bayesian inference. All right, so previous studies have shown that shape discrimination can be decoded from V4 population responses. And we simplified this problem of this decoding problem to compare the responses of two shape-selective V4 units. So one that responds strongly to shape A and the other that responds strongly to shape B. So when the system is presented with an occluded version of A, then what happens on the response state space of the shape A selective neurons and shape B selective neurons is that the neural responses of the shape A selective neurons are high, but shape B selective neurons do not respond very strongly to the presented unoccluded shape A, and therefore the animal can easily conclude that what it's seeing is shape A. However, when the system is presented with a partially occluded version of shape A, then shape A selective neurons respond weakly, and shape B selective neurons also respond weakly, and therefore on the response state space, the responses are very close to this unity line. And therefore, it's really hard to decide what shape the animal is seeing, and therefore, this obscures the shape identity. And our hypothesis is that the top-down predictions on V4 coming from prefrontal cortex reshapes this V4 response inference by moving these initial responses away from the unity line, and therefore it can improve the discriminability of the occluded shape. Right. So in our hierarchical Bayesian framework, the optimal responses are found by maximizing this posterior probability given the sensor input, which is this, so here is the optimal representation, and by maximizing this posterior probability of the neural responses given the input image is equivalent to maximizing the product of these two by Bayes' theorem. So the first part here is represents the bottom observation. So this is the likelihood of the V4 responses given the input image. And then the second part here represents the top-down predictions coming from PFC, because this is a conditional probability of the V4 responses given the PFC responses. All right. And maximizing this conditional probability is equivalent to minimizing minus log of that, which is our cost function E. And if we assume everything to be Gaussian, and this cost function is equal to these two parts. So this first part represents the bottom of observation. So how the inference on the V4 responses given the bottom observation of the input stimuli, and then the second term represents the influence of the predictions coming from PFC on shaping V4 responses. Okay. And we interpreted the initial peak of V4 responses that we saw in the experiments as purely feed-forward representation. And therefore, we model this initial peak in the V4 responses by minimizing just the first term of the cost function, which corresponds to the firing rates that are most likely given the shape and the occlusion level, so the input image mu. And the second peak of the V4 responses was hypothesized as the optimal representation of the neural responses reshaped by these feedback signals coming from PFC, which carries the predictions. And therefore, the firing rates are found by minimizing this entire cost function that consists of the feed-forward signals and the feedback signals coming from PFC. And therefore, the neural responses obtained here integrate the bottom of observation and the top-down predictions coming from PFC. And by using this predictive coding model of V4 and PFC and optimizing the cost function, we can obtain the most likely V4 and PFC neural responses. And so here is a model prediction of the optimal PFC responses. What we see here is that our model predicts that the PFC responses increases with added occlusion, as we saw in the experiments. So it agrees with the experimental observation. But what's more interesting here is the most likely V4 responses predicted by our coding model. So during the first peak of V4 responses, which we obtained by minimizing just the first term in the cost function, which reflects only the inference made by the bottom-up sensory input, we see that the response goes down rapidly with added occlusion. However, that decreases a lot more gradual during the delayed peak of V4 responses, which was obtained by minimizing the entire cost function, which includes both the first term, which reflects the bottom observation, and the second term in the cost function, which reflects the top-down predictions coming from PFC. So based on the optimal model responses, we generated this noisy population of these V4 neurons with the average value at the optimal representation shown in the previous slide, and projected these responses onto the state space of the shape A selective V4 responses and shape B selective V4 neuronal responses. And during the initial responses, so when we were just optimizing the first term in the cost function, which only has the bottom observation of the sensory inputs, we see that when there is a lot of occlusion, which are represented by these green dots, the responses are very close to the unity line. And because of the noise, sometimes it's above the unity line, and therefore it obscures the identity of the shape. However, during the delayed responses, when we included the feedback predictions coming from PFC in our cost function, then we see that the responses are squeezed away from the unity line, even under high occlusion, and therefore, even under partial occlusion, high degrees of partial occlusion, the identity of the shape is so this disambiguates the identity of the shape in V4. So that gives us some idea of how the hierarchical structure of the cortical network can aid in can implicate in terms of computation. Finally, I know I am out of time, but I just want to mention that we are currently looking into dissecting predictive coding in mouse visual cortex. In the first part, we showed that the mouse cortical network is also hierarchy. So maybe the predictive coding, maybe hierarchical predictive coding happens in the mouse visual cortex as well as in the primate cortex. Okay. And so what we want to do here is to see whether there is an experimental evidence for predictive coding in the mouse visual cortex, and if so, how it's implemented in the cortical microcircuit. Namely, is there a specific location in terms of area, cortical area, or cortical layer where the prediction error signals are maybe it's computed at layer 2-3 because that's where it receives both the feedback connections and feed-forward connections, or maybe it's encoded in 5-6 because that's also where it receives both the feedback connections and also this indirect feedback signals. And to do that, so this is actually done through the OpenScope project at the Allen Institute. So the OpenScope at the Allen Institute invites experimental proposals to use their standardized experimental pipeline at the Allen Institute. So this was a very nice opportunity to test the predictive coding theory experimentally in mouse cortex. So the experimental design is to use two-photon measurement in hierarchically related visual areas, namely primary visual cortex, BIS-PM, and retrospolar cortex at multiple depths to understand different layers in the mouse visual cortex, while the mouse is passively viewing the sequence of images. And then the expectation violation was introduced by replacing the last image of the sequence by one of the oddballs. Yeah, and by doing so, we actually found some interesting characteristics of the predictive coding in mouse visual cortex. So we saw there are indeed neurons that encode OPL, the expectation violation. But these OPL responses are image specific, and they are not really some general expectation violation signals. So it responds, so this is for one neuron. So this responds strongly to one oddball images, but not very much to other oddball images. So they are image specific, not just some generic oddball not general expectation violation signals. And another thing we saw is that the error signals are not really localized into one area or one. You actually see that the distribution of the oddball signals are kind of uniform across all different layers and different. But because I'm running out of time, I will skip the next part. So just to briefly summarize, we see that, so like if you look into the single cell responses, we didn't really see much difference between different brain regions. But that was not the case when we are looking into population decoding, because we could actually the decoding accuracy is pretty high for the first two regions, but that was not the case for retrosplenial cortex. But in all three regions, we could actually obtain very high decoding accuracy in terms of context, whether it was presented in the sequence or in randomized control. And this was done in collaboration with David Rurick and Luca Mascotto at University of Oregon. But to summarize, hierarchy of the brain network can be constructed from a detailed network representation of the brain with layer-specific connectivity. And the structural hierarchy implicates hierarchical computation in the brain network, which assigns different computational roles to feed-forward and feedback signals. And in particular, for the feedback signals, our hypothesis is that it actually carries the signals about the predictions coming from the higher cortical areas. And our study supports that the feedback predictions are involved in robust encoding of noisy visual signals. And finally, our ongoing study shows that error signals are specific, and they are present across all cortical hierarchy, across the cortical hierarchy in all layers in mouse visual cortex. And finally, I would like to acknowledge my funding sources and my first, and also I am currently hiring graduate and postdocs, so if you're interested in what we're doing, please email me. And and that's all. Thank you.
This is the sound of one person clapping, so there are many who are doing so. Great, thank you, Hannah. That was a great talk. So we'll start with questions. I'd like, again, the first question to be from a student or a postdoc. So if there's a student or a postdoc who would like to give a question, we'll actually sit here and give a little bit of time for that. Sometimes the silence can get a little awkward, but we would like to have the students and the postdocs first. So you can post it in chat or unmute and ask a question.
I don't think I can view the chat window.
I'll watch it for you.
Yeah, awesome, great.
I have a question, if you don't mind. Great talk. I was interested though about the timing aspect of PFC back to V4. I'm a bit puzzled how PFC is receiving signals fast enough to modulate V4 activity, and I was wondering if you could speak more on that.
Okay, so you're saying that it might be, like, you're thinking of some other candidates, such as something that's in between. Is that the question?
Yeah, because there seems to be like a pretty strong sensory evoked response that's about as fast as V4, and it's like, and I'm curious what's being received and how that's being received faster to then feed back into V4 to modulate. course of
Yeah,
discussion.
yeah. It's a really great question. Yeah. So in terms of timing, so of course another like really good candidate is IT. But the thing is, if you actually look into the temporal scale of the IT responses, it's actually slower than PFC. So PFC like matches better in terms of like as a potential candidate for providing feedback inputs to V4 and providing the second peak in the V4 responses. And the more direct test to see whether actually the second peak in the V4 is actually induced by the feedback coming from PFC is the cooling experiments, right? So where you cool when you cool PFC and then whether you see the second peak in the V4 or not, whether you can still see the second peak in the V4 responses or not. So that experiment hasn't been done like officially, but like the preliminary result actually shows that it's actually the case. So the second peak in this seems to be induced by the feedback coming from PFC.
Thank you.
Mm-hmm.
Great, thank you, and thank you, Joseph, for volunteering a first question. See, if you just give a little bit of time, we get great questions. So now we can open up the floor to anyone, if anyone else has questions. I haven't seen one come into the chat yet, or folks can also unmute and ask the question. I had a doing science question, Hannah, which is that opportunity to use the Allen Institute systems to do a lot of to do experiments is really cool. Do you, in your experience, did you collaborate with an experimentalist to design that experiment, or can the theorists now go in and just sort of bypass all of us experimentalists and just get it all done themselves?
Oh, yeah. I mean, like, if it was just me, I probably wouldn't have been able to do that. But my collaborators include Marina Garrett from Allen Institute. So she knows all the experiments. So, like, we collaborated together. Yeah, and then they also just got NIH funding to continue open scope. So if any of you are interested, submit a proposal.
Excellent. Yeah, that's good encouragement for all of us. Thanks for
Awesome.
that. Zijing Wu in the chat has asked a clarification question. Is the hierarchical structure mainly found in the ventral visual stream?
Not just ventral, like, yeah, it happens in the dorsal. But because I wanted to focus on this one example of discriminating two different shapes under noise, I focused on the ventral. Good question.
And I had a technical question too, Hannah. Now to go with my doing science question. In the second part of your talk, where the decomposition that you had to pull apart the two components of, I think relied on this assumption of the Gaussian noise that you had in there. And I totally understand. I'm not going to get into like questioning people's assumptions because you got to start somewhere, and that's a perfectly reasonable place to start. But I wonder how robust your conclusions are to if noise is different, especially if it's like signal dependent, and now you might not have that. I didn't quite follow the derivation, but I think then you would have some potential bias towards your PFC pathway.
Oh yeah, 100%. Yeah. So yeah. So this is very, very in a very idealized situation where everything is Gaussian, and also you're assuming flat prior on so like you can probably see that there if you I am actually assuming a flat prior on PFC responses and also on images. So of course, that may not be true in the real situation. Maybe it's a little, they may not be flat, and maybe maybe this probability distribution is not Gaussian. In that case, we won't be able to actually do do this derivation for cost function.
Right.
But I think like overall, so this is just it's just like a very, very wild hypothesis. But overall, I think the qualitative results showing that the feedback responses actually reshape the V4 responses so that and then make it more robust to noise. I think that will remain the same, even though it's not perfectly Gaussian, for example.
So there's a question from guest in the chat. Maybe you said this, but how sensitive or variable is the hierarchy index to the efficiency of transduction of the starter cells in the viral tracing?
Um, could you repeat the second part of the question again?
Yeah, so they're saying that the efficiency of transduction of the starter cells when you do the viral tracing can be variable, I guess. And so the guest is wondering how sensitive or variable is your hierarchy index to potential fluctuations. I'm taking it in the experiment based on the efficiency of transduction of the viral tracing.
Yeah. I am not exactly sure, but the thing is, this remains to be pretty robust because so we tested a few different things to test the robustness. So what we basically bootstrap on this connection data. So just like basically looking into the partial, like just the part of the connection data and construct the hierarchy. Or like this, or like just like collecting this part and or that part by bootstrapping, and then we actually got pretty stable and robust hierarchy of the mouse brain regions. So we believe that this method is like pretty robust to a lot of different perturbations.
Great. All right, folks, it's getting on to 12:20, so we should finish up. I just want to thank Hannah again. Thank you, Hannah. And please join us next week. Next week, we will have Jing Shu from the University of Georgia. We're expanding out, making our Georgia community even broader. So she'll be giving a talk in our seminar series next week. So please join us then. Thanks, everyone.
Thank you.
Thanks, Hannah.